Welcome everyone to CanCOVID speaker series. And my name is Susan Law. I'm the Managing Director and the Knowledge Mobilization Lead for CanCOVID, and I'm your moderator for today. It's a great pleasure to welcome Professor Marc-André Langlois, who is the Executive Director of the new CIHR-funded CoVaRR-Net. He is a Canada Research Chair in Molecular Virology and Intrinsic Community, and he's also a member of the College of the Royal Society of Canada. So just a few notes before we get started. Please mute your microphones during the presentation. And we have a question and answer period at the end, and you will be most welcome to post your questions in the chat, raise a hand, just let us know that you have a question. And we definitely want to ensure that these are engaging, so hold your questions to the end and we will have some time for that. I'm delighted now to hand over to Professor Langlois to present your new work.
All right. Well, thank you for that introduction. Thank you for having me here. My name is Marc-André Langlois. I'm the Executive Director of CoVaRR-Net, which is the new kid on the network block. So CoVaRR-Net is the Coronavirus Variants Rapid Response Network. It's a CIHR initiative. So it's born from CIHR funding, but it's part also of the broader Government of Canada's response to the COVID-19 pandemic. The funding for this network is really dedicated to respond to the current stage of the pandemic, which is basically the emergence of all these variants of concerns, which are genetic mutants of the original strain. These variants can cause several problems, including vaccine resistance, immune resistance, and therefore need to be looked at very carefully. So this funding opportunity is really to focus on the emergence of these variants of concern and to define their biological relevance, as I mentioned. So this is a very new network. The notice of decision was on March 23rd, 2021. And as with everything research related, by the time we received the funding, the notice of decision, we were already 1 month behind in terms of getting the network up and running. The funding was available from March 1st. So this is a very urgent and high priority project for a lot of us. The total funding for the network is $9 million. This $9 million is for 1 year of funding. Hopefully, it will be recognized that this is a high value network that is really enhancing Canadian research. And hopefully there will be an extension to that. $6 million of the total $9 million is dedicated for network operation and capacity building. Capacity building is basically generating all the tools we need, collecting all the strains, putting together libraries of expression vectors for the various antigens. So we really want this network to house tools and reagents for the study of SARS-CoV-2 and variants. Also, of that $9 million, $3 million are dedicated for variant of concern rapid research funding. So the network will have this pool of money that it can distribute through competitions, grant competitions, but these will be rapid grant competitions. The way we imagine that is that the proposals will be extremely short, maybe 1 page, 2 pages, and we will attempt to have a turnaround rate for the notice of decision within a week or 2. So it is really a network designed for rapid response, and I think that is the priority. The overreaching goal of CoVaRR-Net, I think why it was designed in the first place, is really to bring together all Canadian researchers for the common goal of understanding and stopping the SARS-CoV-2 pandemic. And here I've really underlined and highlighted the word all, is that this is really an opportunity to bring together both academic researchers, government researchers, and researchers of every type, whether you are basic, clinical, or applied. I think one thing that the pandemic has done is made us all realize how it's important to work together. We cannot just work separately. pandemics affect everyone in society, and all the researchers of all fields are required to make a joint effort, to create this joint effort to try to understand how to stop this pandemic, how to control it, and how to also predict where it's going in the future. So our vision and our mission at CoVaRR-Net is to rapidly and efficiently act on the emergence of new SARS-CoV-2 variants of concern. And there are three primary goals here. First is to functionally characterize the variants of concern's features. This means including the potential for vaccine resistance, breakthrough infections, and immune escape. So this is a lot of this is laboratory work. We're also, the network is also focusing on trying to predict the evolutionary trajectories of the variants of concern. And this is done in vitro, in vivo, in BSL-3 labs, and also in silico. We know that when SARS-CoV-2 replicates, it makes errors in the replication process. And these errors, these mutations are basically what are driving the emergence of these variants of concern. So as long as the virus is given an opportunity to replicate anywhere around the globe, we will see variants emerge continuously. And the third objective of the network, and this is a very important one, is that it is also designed to rapid communication of information in real time. The knowledge mobilization component of the network is extremely important. It's not enough to do experiments in the lab and to wait till the data is published. We want this information to get out as quickly as possible to Canadian public health officials, decision makers, but as well the broader international scientific community. So the network is really there to serve the purpose of blunting the course of this pandemic and sharing this knowledge very, very quickly and efficiently. So how is CoVaRR-Net, what is the architecture of CoVaRR-Net? It has two major sections to it. There is a basic science section and an applied science section. The basic science section has 5 pillars. Pillar 1 is immunology and vaccine protection. Pillar 2 is in vitro and vivo characterizations. Pillar 3 is functional genomics and structure function of the VOCs. Pillar 4, viral genomics and sequencing. And Pillar 5, in silico modeling and computational biology. So these are more the lab-based pillars. And then there are the applied science pillars. So public health, health systems, and social policy impacts. Indigenous outreach, there's a full pillar that is dedicated to Indigenous outreach to ensure that if variants of concern do emerge in Indigenous communities, we have someone there to liaise with the network and those communities. And then there are the cross-cutting pillars of CoVaRR-Net. Pillar 8 is the knowledge mobilization pillar. And this pillar here interacts with all the other components of the network. As soon as there's new information that is generated in any of the pillars, this information is funneled into the knowledge mobilization pillar, which will then reach out and communicate that information the most efficiently as possible. Also in CoVaRR-Net, there is a very important component in regards to equity, diversity, and inclusivity. The EDI director sits on the executive committee and provides a complete network oversight. Considering this SARS-CoV-2 or COVID-19 pandemic, it affects everyone across the globe. And we want to make sure that the research that is conducted is also very inclusive and reflects all possible components and communities in society. So there is a very close network oversight by the EDI director. And finally, the last component of the network is the central administration of the network. This is where shared resources are located, data sharing, the biobank. We have an ethics director and a citizen and patient outreach. An important component for the network is resource sharing, data sharing, and biobank. If we want this to be a rapid response network, we have to be able to share reagents very quickly, share viral isolates, share plasmids, expression vectors, share antigens. And one of the best, most efficient ways that we've imagined to do that is to be able to house these shared resources internally and be able to dispatch all of these reagents very quickly to the members of the network. So the complete network, all the standing members, regular standing members of the network, there are 40 individuals currently in the network. There is an executive committee composed of 10 members. So these are the pillar leads, the 8 pillar leads, the EDI director, and the executive director. These individuals are the ones making the rapid decisions regarding the network. These individuals, especially the pillar leads, are supported by experts in their fields. These are called deputies in our network. Each pillar lead is supported by 3 to 5 deputies that provide advice, that provide support, provide expertise to each of these pillar leads to accomplish various tasks. And we'll see a little bit more about what these tasks are in a few slides. Then there are the 5 directors for EDI, that sits on the executive, ethics, biobank, data sharing, comms, and community outreach. 2 of these directors are supported by associated directors, that's at the biobank and for data sharing. And then we have our knowledge users, our patient participants. There are 2 of them that are part of the network. So this is a total of 40. So who are these, the key people in the network? Unfortunately, I cannot name all 40 right here on the slide, but I will name the pillar leads. So for the immunology and vaccine protection pillar, the pillar lead is Jen Gummerman at the University of Toronto. In vitro and vivo characterizations, Louis Flamand at the Université Laval. Functional genomics and structure function of VOCs, Anne-Claude Gingras at Sine Health Systems, Lunenfeld in Toronto. Viral genomics and sequencing, Yanis Ragusis at McGill. In silico modeling and computational biology, Jesse Shapiro at McGill. Public health, health systems, and social policy impacts, Nazim Muharrejeen at U Saskatchewan. Indigenous outreach and liaison, Michelle Johnson Jennings at the University of Saskatchewan. And knowledge mobilization, the pillar lead is Jeremy Grimshaw at the Ottawa Hospital Research Institute. And our EDI director is Nainan Abraham at UBC. There's also our directors. Our directors are Fatima, who is in charge of media outreach. Biobank is Angela Crawley at OHRI as well. Data sharing, Amy Shu at the Briere Research Institute here in Ottawa, and bioethics, Ray Saganer, who is located at the Ottawa Hospital. There we go. So what is the reporting structure of this network? So CoVaRR-Net interacts very closely with a leadership group. This leadership group is chaired by the Deputy Minister of Health, Stephen Lucas, and a number of other individuals that are involved in variant of concern surveillance, sequencing. There's also the NML that is involved in that, and the BCCDC. So there are a number of individuals that basically provide CoVaRR-Net with marching orders, and CoVaRR-Net also reports to. Then there's, to the leadership group, there's a Science Advisory Council chaired by Alan Bernstein, and a number of experts in the field that provide scientific advice to the leadership group to help focus the research efforts of CoVaRR-Net. And CoVaRR-Net itself is supported by an science advisory group. This is a work in progress. This advisory group is chaired by Matthew Miller from McMaster University. The composition of this group is currently underway, and this group will help CoVaRR-Net identify subjects of interest, of priority, and will also provide advice as to what should be funded in terms of rapid research response. So overall, how does this work? How do we decide as a network what to work on? Well, there are several sources of input in regards to what should be analyzed, what variants of interest or variants of concern should be analyzed. This information can come from the leadership group. It can come from the Public Health Agency of Canada that sends a request for VOC testing to CoVaRR-Net. But there are other sources of input equally where CoVaRR-Net can self-initiate some testing. If CoVaRR-Net identifies variants of interest or of concern that require testing or high priority testing, it can do so in its own right. Then there are other entities that can provide requests for testing. There are the various Canadian sequencing effort. We can think of CanCOVID, public health, and also the wastewater surveillance network with which CoVaRR-Net is trying to build a close relationship. I think we've all come to realize how important surveillance is at the wastewater level. It helps predict where we're going and what is coming next. And this is a very important source of information for CoVaRR-Net, is to understand what is happening in the wastewater, because we usually see whatever comes up in the wastewater is reflected in the population after 7 to 9 days. So it is a good warning system for what is coming next in terms of variants. And of course, there are the variants that are identified in public sequencing databases. So all of these inputs come to CoVaRR-Net, and CoVaRR-Net prioritizes and vets the various requests for testing. There's going to be a process within CoVaRR-Net to make that decision, because of course testing is costly and cannot just functionally test every variant of concern coming to us. So there's a decision that needs to be made. And then the testing will go to each of the pillar leads. And the pillar leads will basically then connect with their own sub-networks, with their deputies, and basically dispatch requests for testing according to the specialties of each pillar. So who are the labs that each pillar will be connecting to? Well, they will be the labs of the deputies of the network, but also all of the Canadian labs working on SARS-CoV-2 and COVID-19. So this is a very open network. It's not a closed ecosystem. All the testing and the research is not all done within the network. We see all of the Canadian landscape as partners of CoVaRR-Net. And what we want here is, when we perform the testing, we will identify the specialized labs with the right expertise, with the right bandwidth to perform these testing requests efficiently, and financially support these labs to carry out this testing. So these can be any of the Canadian labs, research labs, and also the CIHR variants of concern stream 2 networks that were established a few weeks ago. So what CoVaRR-Net wants to do is really bring together all of the Canadian landscape, not exclude anyone, and really seek out the best labs to get that information as quickly as possible. To be able to carry out this research, the biobank is critical. The biobank will be able to provide reagents and whatever other material supports to these labs, and as well any sort of strain isolation, variant isolation that occurs within the network will also be fed back into the biobank. Then once the results for the testing are acquired and we have that information goes immediately back to the network. What kind of information? This can be serology, antibody resistance assays, immune T-cell assays, infectivity, in vitro, in vivo assays, you name it. Each pillar will be responsible for acquiring their specific set of information, bringing it back to the network. And this is where our knowledge mobilization pillar comes into action and basically reaches out to inform public health and government decision makers. And of course, as I mentioned earlier, CoVaRR-Net will be also hosting several competitions for rapid research grants. with our $3 million envelope. The Canadian Immunity Task Force as well has, well, there's a partnership between CoVaRR-Net and the Canadian Immunity Task Force that is being established right now to see if this is something we can do jointly. So a lot of ramifications. More specifically now, examples of what can be done. If we take, for example, two examples here because of the time, let's focus on in vitro and vivo characterization and knowledge mobilization. So how would that work? So pillar 2, which is led by Louis Flamand, is the pillar for in vitro and in vivo characterization. Louis is supported by 4 deputies, Jennifer Corcoran, Sylvia Vidal, Denis Leclerc, and Stephen Barr. And these are individuals that are actively involved in research that requires biocontainment level 3 and animal studies with SARS-CoV-2 and variants. So a request for testing would go to pillar 2. And then, for example, the variant of interest that is circulating right now, the B11617, then the pillar would assess what kind of information or materials are provided. Are swabs provided? If so, well, what the pillar will try to do is isolate the virus from these swabs and make viral stocks. So they will connect with various stream 2 networks or other Canadian laboratories that have BSL-3 capacity, and they will carry out the isolation, the in vitro characterizations, the in vivo characterizations. If this information is just sequence only, and this often happens, we don't have the isolates, we don't have the samples, but we need to test. We also have pillars that can provide support, such as the functional genomics pillar, to help produce antigens, or even through pillar 2, perform reverse genetics to create an infectious replicative strain of the virus. Again, all of the testing will be coordinated by this pillar, reaching out to Canadian labs with the right bandwidth and capability. And ultimately, all of this information basically streams back to various pillars of the network. For example, the viral genomics pillar to get full sequences of the variant. If we do get simply a swab, in silico pillars to do the computational biology. Ultimately, all of this information comes back to CoVaRR-Net, and then it gets mobilized. So this is an example of how pillar 2 would work. An example of how pillar 8 works, knowledge mobilization. Well, the pillar mission is really to undertake a state-of-the-art knowledge mobilization activities to ensure that Canadian citizens, healthcare professionals, public health, health system policy leaders, and researchers are informed in a timely fashion about VOCs. Here's an example of how this pillar could work. Again, let's take the current variant of interest, the 617. We could go forward to the pillar 8 and ask a very simple question. Is this new variant a problem for current vaccines? And what this pillar would do in this instance is provide an evidence mapping strategy to provide answers to that question. So it would search for existing evidence in the current literature. If that evidence exists, it will not just simply compile those articles and that information. It will go a step further. It will assess the quality of that information and the relevance to that specific VOC. It will prioritize gaps in the knowledge and identify who can fill in those gaps. Ultimately, what this pillar wants to do is to create a tailored, actionable message to be able to disseminate that. If there's no evidence of what this variant does, then that pillar will try to figure out how can it acquire that information. It can outsource requests for testing, or actually ask the network itself to perform those assays. Again, it's all in the goal of acquiring some quality information that will be assessed and interpreted, and then it will create a tailored, actionable message to engage the proper channels. So these are two examples of how the network works. What are the current priorities of the network? Where are we at right now? We are 1 month in from receiving the notice of funding. The network is currently hiring its staff and its coordinators. We are building the website. The website will be very important to us. This website will be directed to the general public and the media. I think several of the researchers of the network get regular requests to speak to the media regarding various issues. And we really want a home base to provide that core information to the general public in a very clear language to get the messages across about what is important, what is to worry about, what is not to worry about in regards to variants of concern, or even the vaccines coming out, whether they're going to be working on these variants or not. So we want to get that message out clear to the public. CoVaRR-Net is currently reaching out to other networks. Amongst all of these other networks are 2 other CIHR funded networks, the COVID-19 Clinical Trials Network. There's also REMAP-CAP, another clinical trials network, an international network, and also the Canadian Immunization Research Network. So we're trying to build this broader landscape to streamline and to bring together Canadian research for this common purpose. We also want to integrate and create partnerships with the wastewater VOC surveillance network. As I mentioned previously, it is very important for CoVaRR-Net to build this relationship with them because there's extreme value there to see what's coming next, which is part of our mandate, is to be rapid response. Well, if we want to be rapid response, we have to know what's coming. Reaching out to Canadian drug and vaccine companies, we have this tremendous capacity within the network. And therefore, there are opportunities there for us to shoulder some of the research that is required to bring drugs and vaccines that are made in Canada to the market. So we are offering our support there, reaching out to public health labs who are overwhelmed with trying to isolate all of these variants of concerns. We want to offer our help to contribute to that effort. And to be able to do that properly, CoVaRR-Net is trying to establish a consortium of Canadian biosafety level 3 laboratories and ensure that within this consortium, there are some standard operating procedures that are established, SOPs, to ensure that all the experiments and the testing that is done with and between network members is standardized. And standardized within the network, and also standardized with how those experiments and those isolation procedures are done in the public health labs. So this is a work in progress. I mentioned the biobank several times. This is extremely important for a rapid response within the network. Our biobank will also be reaching out to other biobanks. We're not isolated. We want to work collaboratively. So we will be reaching out to other biobanks as well to see how we can all work together efficiently and quickly. And then the final point I want to make before I end here is the current research priority of the network is really to focus on genetic evolution of variants of concern. Can we predict and preemptively test what is coming next? We know these variants are evolving, they're acquiring new mutations. Can we predict these genetic evolutionary pathways, trajectories? Can we test preemptively new variants of concern that have not emerged yet, but are poised to emerge? So these are the sort of questions CoVaRR-Net is asking itself. So that concludes my talk. Thank you everyone for being here.
Thanks very much, Professor Langlois. That was an excellent overview of a very complicated and exciting new initiative. We have a few questions and we'll just take a couple of minutes here. We are supposed to end at 4:30, but let's just go a couple minutes over. The first is, will the network be collecting samples from defined populations so that you can monitor VOC penetration, regional variation, etc.?
So there are several strategies to bring samples into the biobank. There are several studies that allow for secondary use of samples, several studies that allow for banking of samples. My lab and the laboratory of Anne-Claude Gingras in Toronto are doing a very large volume of serological tests using high throughput instrumentation. And through our labs, we help support numerous studies across Canada. And several of these studies do allow for banking of their samples and secondary use. So we are collecting some of those samples that we're allowed to. The biobank will also have a different phase where we will do active recruitments for large blood draws for more complex T-cell studies that require larger volumes. So there are several strategies going on at the same time to create this VOC biobank.
Thank you. We have a hand up from Richard Leduc. Would you like to speak up with your question?
Oh, yeah, I was just about to write it, but fine, yeah, I can ask the question. I've got a couple. One, practically, how is it best to submit our applications for funding? Is it through ResearchNet or is it through your website? That's my first question. My second question is, how will it work if we, for instance, if we have a drug therapeutic that potentially help? How will CoVaRR-Net help us to reach out to pharma or VCs or anybody else that could help us go further in characterizing these molecules?
No, these are the things we're thinking about all the time. So for the funding, first thing, there will be a call for proposals once CoVaRR-Net has all its staff, because I think everyone can appreciate how resource intensive it is to carry out a call for proposals, for research proposals. So there will be a call that will be made through the CoVaRR-Net website once we go live, once we have the capacity. And then we will, there will be a certain number of focused topics that we believe are priorities. And then we invite all of Canadian labs to apply to this. So these will be rapid research grants. They are small in scope and they're meant to get information out very quickly. In regards to new drugs, new compounds, anything that can benefit the Canadian effort to get drugs to the market, well, CoVaRR-Net is trying to establish those relationships with the companies, with the clinical trials network. So once these relationships are formalized and we start building these relationships, when a researcher comes to CoVaRR-Net and says, well, we have a molecule, a small molecule, for example, that is interesting, that blocks entry or reduces pathogenesis, then we will be able to put those researchers in touch with the right people. This is the dream. This is what we're working towards right now, is to have this network, this relationship, this good relationship with industry and clinical trial networks.
Merci, Marc-André.
There we go. Sorry, I muted myself. Just a last comment and question is just related to that in the industry relationships there. The comment is that connections to industry and vaccine developers may not be that clear, and IP protection issues may arise that are not clear. And it's critical to define correlates of immune protection and how this informs the decision-making tree. Do you have a response to that?
Well, those are three very complex topics. We've been up and running just for less than 30 days, so everything's a blueprint right now. What we can imagine, so everything is right now is a dream, is a goal, and we'll have to figure out the details of how all of that will work. But ultimately, what we're dreaming about with the industry is that, for example, a company has a compound and they request animal testing, for example. Well, through CoVaRR-Net, there are several labs that have BSL-3 facilities associated with animal care facilities. We could put them in touch and contact and create agreements for testing. Of course, IP has to be protected. Of course, there will be contracts. This is absolutely obvious. But CoVaRR-Net is not there to benefit from this. We are there to help the Canadian landscape. We want to get these drugs to market. I think Canada needs a win in this pandemic in terms of getting a vaccine that is Canadian made. out to market, get some Canadian-made drugs out to market. This is the message that the government of Canada has given CoVaRR-Net, is that we need a win. We need to bring all of our research together, our industry and our academics, to work together towards this pandemic. So that is the dream, and we will try to navigate those IP landmines, but of course it is complex. And critical to the correlates of immunity, of course, immunity is not just antibodies, and we know that. So there are very experienced teams working on T-cell studies, cellular immunity, ADCC. So there are various innate immunity. There are various components to the immune system and to our protection to the virus. And all of these labs are being brought in as experts within CoVaRR-Net, and we will try to dissect all of these correlates in due time as best we can.
Thank you very much. I'll have to end it there. We have gone over time, so apologies for that. There's still some outstanding, excellent questions. Would you suggest that they be directed to you, Professor Langlois?
I'm happy to answer them offline through email. Absolutely.
That's fantastic. Well, thank you very much, everyone, for joining. Next week, we'll hear from the scientific director from the CIHR Institute of Neurosciences, Mental Health and Addiction, Dr. Samuel Weiss, who's going to talk about their major funding initiative on mental health and the systematic reviews they've inspired there. So thank you so much, everyone, for joining us, and take care. Thank you, Professor Langlois.